celebrate.
Happy Wednesday.
You should have given me half point on that.
That wasn't very enthusiastic. Make sure that everyone gets their homework turned in. We're picking that up after class. I was just talking to the students down front. The big wave competition is happening tomorrow, so not conflicting with the exam. Everyone can go out to that and come back to study and be ready for the exam Friday morning. Today, I'm going to go over some points of confusion that I know students have been emailing me about so that everyone's on the same page as far as what I expect, particularly polypeptides and how to draw them. We'll do some translation problems to make sure that everyone understands the questions that were on the homework and what to look for in those translation problems. So we're going to do a bunch of those. If we get past that, there are a few more things about translation to cover. If not, wherever we end today, that up until that point is fair game for the exam. All right. So the normal announcements to start off with. Last day for final point adjustments on the first exam. So if you haven't, come and talk to me if your exam. Now's a good time to do that. You've got until the end of the day. Come and talk to me. I have office hours 9:30 to 10:30. If you can't make that, send me an email. I'll find another time to meet. Please come and talk to me if you have issues, okay? The exam score should now be updated on La Lima. I fixed a few things that students have talked to me about. I made sure I'm doing my best to make sure everything gets updated. If you talk to your TI after I updated on La Lima, I'm still getting those scores input, but I will get to it hopefully before Friday and when we take the second exam. All right, just like last time, but it's going to take us a week or so to get those second exams graded. Once I get the second exam scores, I'll post everything on La Lima up until that point. All the current homework, all the current quizzes, and the second exam grade for you to look at, make sure we've tracked everything properly. We haven't made a mistake in entering a grade somewhere. All right? Help us make sure that we're not making mistakes and keep track of your own scores, make sure that they're entered properly. All right. Second exam is on Friday. Hopefully, a lot of you could make the review session last night. How did that go? Well, good. So you have 2 more days to prep. You've got study guides, you've got mastering biology, you've got access to your TIs. At least one student has written and asked me about the homework key for the homework you just turned in. All right, so this is the issue being that as soon as I give the key out, I can no longer accept late homework. So normally I accept homework through Friday. All right, I want you to have that key to look at. We're going to go over similar problems today in class, so hopefully that'll clarify any questions you have on the homework. I will post the homework key at the end of the day on Thursday. Or by evening, I mean at the end of my work day when I go home, I'll post it. Once I post that homework key, I won't be accepting any more late homework. All right, so you need to get it to me before then if you haven't turned it in already. All right, so that's hopefully a reasonable compromise to give those who haven't turned it in yet a day to sort of get it in late and still get you the key in time to look it over before the exam. All right, because our exam is on Friday, I've canceled recitations for this week. However, because you have amazing TIs and they love you and they want you to do well, they're going to use that time on Thursday. All of them are going to come in and out to the and have an open review session where you can go in during the normal recitation period. There'll be some combination of TIs available for last-minute review questions. You're still confused about a concept. All right, they're making themselves available to you all day on Thursday during those normal recitation periods. That's sort of an open review time. All right, so those are the normal times and places of recitation. Like I said, there'll be some combination of TIs available at those times and places tomorrow. Just be aware that those rooms, as you know, since you are in those recitation sections, they can only hold so many people, somewhere from about 30 to 45, I think is the largest room. So if you show up and the room is full, don't cram 100 people in there. You might have to wait a minute for someone to get a question answer and leave before you can go in. All right, but I think that there are enough times throughout the day that won't happen. So your TIs are amazing, and you should thank them for that when you see them, right? Because they're going to be available pretty much all day on Thursday if you want to go and talk to them about final questions. Okay? Any questions about the review sessions that are going to be held tomorrow, the exam, material? Everyone's got it all covered and figured out? Ready for the exam on Friday? All right, so let's start off today by going over a few things just to clarify so that everyone understands what I'm looking for when I'm asking some of these questions. All right, so first of all, what is this a representation of? The central dogma. All right, so there's another representation of it. Replication, translation, transcription. All right, everyone should be familiar with that. Be able to draw that in some form. I've also gotten a lot of questions from students about how exactly to draw the ends of polypeptides. All right, and so I want to clarify that so everyone's on the same page. Because the polypeptide has directionality, got an amino terminus and a carboxy terminus. You draw the structure, you have to show me those ends, and they should be charged. Right, we're drawing this as in physiological conditions in the cell. We're going to have the charges on the ends of those polypeptides. Also, for drawing the polypeptides, there's been a lot of questions about whether or not you can use skeletal structures. I will accept skeletal structures. However, you can't get sloppy. It has to be clear. Right? Sometimes those lines, your pink lines, get a little questionable at all, and you can't interpret it, and you're going to miss those points. Right? So this way, I feel is much clearer. If you learned a skeletal structure and you want to use that, that's fine. Just make sure you are very clear about your lines. And I want to see the charges. Yeah?
About the hydrogens on the carbons in the area.
Right, so just like we did for the nucleotides, if it's a carbon, I'm going to assume anything that's not shown is a hydrogen. So that saves you some time in drawing it. Feel free to draw the hydrogen. If you want to just draw the carbons, that's fine. All right, just like in the nucleotide carbons, I'm assuming anything that's available as a hydrogen are not shown. For the functional group, show everything. Okay? Any questions on what I expect for that? I'll go through a few more examples. Before I do that, clicker questions. So I know we've been having a lot of technical issues. I've been in contact with iClicker, sent them a lot of emails back and forth. They sent me a software update. Hopefully that's going to fix the problem. We'll give it a try today and see what happens. So let's go ahead and try.
Hey, it's not me, it's the video.
Tell me what those two amino acids are.
No! Are you serious?
Yes.
Canadian.
You think there's a carbon and there's bonds available to use the hydrogen?
Yeah, I made a little bit again when I brought it.
All right. So today is a lot of practice for the exam. You need to know your amino acids. tell me what it is. All right, get your answers in. Let's take a look. See what you think.
It's like, this is what it looks like. I just Googled like a slime.
All right. Seems like you guys are doing all right with your amino acids. All right, so we have 15 serines shown there on that polypeptide. All right, so let's continue talking about a few more details about how to draw your polypeptides. Right, so here is the structural version of that. Here is the, if I just ask you to give me the sequence of amino acids and not to draw the structure, I want to see something like this. What I'm looking for is that you understand which end is the amino terminus and which end is the carboxyl terminus. All right, I want to know that you understand that, so you have to indicate that to me no matter how you draw it. So there's two different versions of the same thing depending on what I would ask for. And then the final thing to clarify regarding drawing structures, we talked a bit about the open reading frame, what we expect if we look at different segments of the open reading frame, and we're going to practice some of that today. All right, but if we're looking at the entire open reading frame, so in this case we have a polypeptide that's 3 amino acids long. So we have that amino terminus, methionine, carboxyl terminus. And that's if we're looking at the entire open reading frame. We know it starts with a methionine, it ends on a carboxyl terminus. What about if we're looking at a stretch of amino acids in the middle of the open reading frame?
You have 3 options.
Sorry?
You have 3 options to read it differently, I guess, depending on what one.
You do. We're going to get to that. Right now, let's just talk about how to represent it in a drawing. You're way ahead. We're still talking about this drawing. All right. So because this is in the middle, you know, it's bonded to other amino acids on either side. We're just looking at a piece of it. Again, I want you to represent, I want to know that you understand which end is which. All right, so indicate that to me in some way, and then you can put dot, 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 wherever you want to indicate that it's connected to, it's part of the middle of a polypeptide. All right, so this is one way to do it. There may be other ways, but as long as it's clear what the order is, that you understand what the terminus is on either side, connected in some way with other amino acids. All right? Is that clear? Are there any questions about what I expect if I ask you to draw or write out the sequence of amino acids? Clear? All right, then let's practice some translation problems. Question? Oh, sorry. Very good question. As a matter of fact, the very next slide goes over that. So this is where we ended. So the question was what to do if you have 2 overhanging base pairs at the end of a sequence that you're trying to translate, what I expect you to do. So we looked at some examples on Monday, but it was sort of at the end of class when things are, everyone's packing up and not really paying attention. So we're going to go over it again. This is where we ended. We had translated this open reading frame. We're at the beginning of the open reading frame, so we know it starts with the methionine. We've gotten through the tyrosine, corrected the position of now. The question is, we've got one overhanging base pair there's no way to know what amino acid is. You've got two overhanging base pairs, you might be able to figure it out. So if it's clear what that amino acid is, just based on the two base pairs, I expect to see it in the translation. All right, so figure it out. What are those two base pairs? Can you tell what that amino acid is or not?
The whole thing. Yeah.
So you can't just shut your brain off at the last triplet codon. You have to look at what's left and evaluate whether or not there's enough information there to tell me what that last amino acid is. All right, let's take a look and see what you think. All right, excellent. So I expect to see that on the exam. If you can't tell what the amino acid is, don't worry about it. If it's clear, it's a single amino acid, write it down. All right, so let's keep going with examples of translation problems that maybe you had questions about on the homework or we haven't run into yet. Remember, you need to read my questions carefully. All right? Read the instructions carefully. Read all the words carefully. So the next question, I'm going to show you a sequence. I want you to tell me how many stop codons are present. There's a sequence. What do you think? All right, everyone count up those stop codons. So I just had a question about, I'll share with everyone. All right, the question was related to whether or not to show the charge on the side chain. Don't worry about that. I don't expect to see that on the side chain. Draw that as it is in the book. All right, I just want to see the charges on the ends of the peptides. Yeah. So that is the thing you have to figure out about this question. Right? What am I asking for here? Yeah. Yeah, carboxyl. So for the side chains, don't worry about charge. Yes. Yes. That's it. So read the question carefully. We're going to go over it, right? So what am I asking? So what does it say in the question? How many stop codons are present? Present. How many are there? Right, I'm going to share show you an example of what this. All right, everyone get their answers in. It says how many stop codons are present in this sequence. All right, let's take a look. All right, so I think the question, right? So some of you interpreted it as asking for which one. You know that it's an open reading frame because I've shown you that. You know that there can only be a single stop codon in an open reading frame. That's not what I asked for, though, right? What I asked for is how many stop codons are in that sequence. How many are there? There are 3. All right, there they are. All right, so that's a different question. How many stop codons do you see in this sequence? In this question. All right, how many in-frame stop codons are there? So I can ask you about the sequence in general, or I can ask you to consider it within the frame of translation. Everyone get their answer in. We'll take a look at that. All right, let's take a look. All right, 1 is correct. So there's a difference in the way the question is asked. I'm asking you for what the translation is, or just the sequence in general. All right, so if we look at that, let's demonstrate the difference one last time. So there are potential stop codons. The first question is, what are the potential stop codons? The second question is, which is the one that's in frame? That's a part of that translation, that's part of that open reading frame. So I gave you that information about where the open reading, that it was a full open reading frame. We expect to see methionine to start with, a stop codon on the end. We find our methionine. We work our way triplet codon. So that UAG is the only in-frame stop codon. There's always only one for a complete open reading frame. All right, and then we can also translate that. I think this is clear, but let me just state it to make sure everyone understands that the stop codon, there's no amino acid for the stop codon. It's simply a signal that says translation ends here, which means that that polypeptide ends at the amino acid just before the stop codon. If you hit the stop codon, nothing after that matters, nothing after that gets translated. Just the end signal. All right? Clear? Any questions? Yes, Haley. You don't represent the stop codon in the polypeptide sequence. All right? If there's no amino acid, there's no part of that represented in the polypeptide. It's simply a signal for the translation machinery to stop. Okay? All right, so we've gone over some of the details about translation and open reading frame. Let's do some problems similar to what you had on your homework. All right, so get together with people near you. Work on the translation for that sequence. Remember, you have to take into account the information that I've given you about where in the mRNA that sequence comes from. Yes. Right. You can write continue, yeah. Yeah, so you can mix and match those in to represent the sequence properly. That's exactly what we talked about at the beginning of class, right? I want to represent that there's a bond there, and you understand that there's sequence on either side. Yeah. All right, has everyone had a chance to translate that? All right, I'm going to switch over to document camera so that we can do it together. This one, I think, is hopefully straightforward. So based on where it is in the open reading frame, what do you expect to see? What does it start with? AUG. Is there a stop codon in the translated sequence? No. So those are your criteria for what the proper translation is. We know that this part of the sequence isn't translated. So we're not starting right at the beginning of the open reading frame. We have to look for that AUG. All right, so we go through our sequence till we find an AUG, and that's where we start. So AUG is methionine. What's the next one? Next one? Next one? Next one? Can we tell what the next one is? No? So what goes on this end? What goes on this end? All right, when you are working with the beginning of the open reading frame, look for that first AUG, translate from there. All right, there's no other possibility of reading frame. Starts at that first methionine. Yeah, sorry. I squeezed it in there. Yeah. No, don't. If there's overhanging base pairs that you can't tell what they are, don't worry about representing them. Yeah. Okay? Clear? So let's look at a slightly more difficult problem. There's the one we just did. Let's move to doing a translation in the middle of the open reading frame. But before we do that, all right, so that's the sequence. Let's think about the relationship of exons and introns to the open reading frame. And what the sequence in the middle of an open reading frame, what information that contains. All right, so if we are splicing two exons together, what's the sequence around that splice junction that ends up in the mRNA? The blue letters squished together, right? Yes. All All right, so you get the ends of those exons put together in the middle of an open reading frame. Is there enough information to tell what that open reading frame is if I give you just that sequence of letters? Talking about the blue letters. All right, so that's what's going to end up in your open reading frame in the mRNA. Can you tell me what the open reading frames for those letters are? Or is? Sorry? Here, I can draw it for you. So this is what our splice site is here. So in the middle of an open reading frame. Can you tell me what it is? There is an option to tell me you don't understand.
Is there a wrong answer to that?
If you don't understand the question, it will not be counted wrong. And then everybody else. Everybody else answers. All right, let's take a look.
So this is like
All right, so definitely you're confused about what I'm asking, right? So basically, oh, what just happened there? Sorry, wrong button. All right, so what I'm asking, right, is if this is the sequence in the middle of the open reading frame, is that where you start? Or do you start there? Or do you start there? Right, there are three possibilities for where to start your triplet codon for the translation. We call that three different reading frames, three different ways to translate that sequence. All right? Try again. Is there enough information there to tell me which one of those is the correct one? All right, we're not at the end where you have the start codon that you expect to see or the stop codon that you expect to see. So can you tell me what the right frame is? Sorry? So the reading frame is the correct one, but there are three possibilities for where to start that reading frame. All right, let's take a look. All right. There isn't enough information in what I gave you to figure out what the correct one is, which means you have to translate all of them when I give you a problem in the middle of the open reading frame. So let's go back to that translation problem that I just showed you. We're in the middle of the open reading frame. So you can't tell which one is the proper way to translate it, so you have to check them all. Translate that.
There's only one
Yeah, you can't use the second one. The second one doesn't work. The first one is all wrong. Oh, you're right. You're right. It's a little bit. The first and second one. Let's try the third one. Why does the second one not work?
I know this is a little bit hard to do when you don't have the sequence in front of you. Right? It's easy to make mistakes when you're looking up and down. We'll do it together. It'll be easier on the exam when you actually have the sequence in front of you.
Okay.
All right, let's take a quick look together. I want to show you one more example before class is over, just to make sure that everyone is clear what you need to do for these problems. All right, so we're in the middle of the open reading frame. We know that there's no information there to tell us what is the proper one of the three possibilities, so we have to check them all. So if we do the first reading frame, right, and I'm not going to translate it, I'm going to trust that you guys can see that. What do we run into there? So that can't be the right translation frame, all right? That's a stop codon. Translation frame 1, not possible. Second one, are there any stop codons there?
No.
All right, so that one's a possibility. Third one, where is the, so what do we run into there? So 3 can't be the possible translation frame. So that means the answer for this question is the second reading frame, and that would be the translation I would expect to see. All right? Tell me 1's not possible, 3's not possible, and then give me the translation for number 2. All right, so just to run through one more, I'll give you a quick look at it. So the quick way to do that is just to do what I just showed you before you go through the trouble on your exam of spending the time to translate everything, because that takes a lot of time, right? Just box out the triplet codons of each reading frame and see if there's a stop codon or not. All right, see if it matches their criteria. That reading frame matches the criteria for the parameters that I've given you. So, take a quick look at that one. Different sequence. Check all 3 reading frames. Alright, so I'm giving you a time-saving tip for the exam. Alright, everyone have a chance to scan through all 3 reading frames? For a quick minute. Alright, let's take a quick look at the reading frame. These will be in the slides that I'll post when I get back to my office. So There are more examples in my lecture than we're going to get through today, so do those as practice for your exam. Go back and look at them, check all the reading frames, do the translations. Good practice problem. All right, so for this one, let's do the same thing. First reading frame. What do we run into? Stop codon, 1 isn't a possibility. Second reading frame. We hit any stop codon? 2 is okay, so you'd have to translate that one. Look at 3. Do we hit any stop codon? So you'd have to translate that one as well. So for this problem, there's not enough information to tell which one is the correct reading frame. You have to give me both. Right? For some of the problems I give you, there may be multiple reading frames that are possibilities. I expect to see all of those that meet the criteria. On the flip side, so we've been looking at the open reading frame, the middle of that, and how to do a translation in the middle of it. What about if we're looking at the end of the open reading frame? All right, what about that problem? You have to write all of the possibilities to meet the criteria. Well, so if there's only one way to translate it that meets the criteria for where it is in the open reading frame, then there's only two. You have to do however many as possible. Yeah. One more thing I want to show you before we end class. All right, so for this one, you know that it is a piece of sequence at the end of the open reading frame. So what do we have to see in that sequence for it to be correct? Stop codon. All right, so this one, reading frame 1. Stop codon. So in this case, reading frame 1 is a possibility. Reading frame 2. Do we see any stop codon? No. So reading frame 2 is not a possibility. We expect to see a stop codon. Reading frame 3. Do we see a stop codon? Yes. Stop codon there. So reading frame 3 is also a possibility. You have to give me both 1 and 3 for a question like that. All right? Is that clear? So you have to look at where The sequence, I tell you the sequence is in the mRNA relative to the open reading frame. Use what you know about that open reading frame to give you the characteristics that you expect to see in that sequence. And then give me all possible translations that meet those criteria.
Yeah.
All right, so the final thing is, all right, so just remember your properties of open reading frames. Remember that open reading frames always start with an AUG. They always end with a stop codon. That stop codon has to be in frame with the AUG. If you're at the beginning or the end, those give you the frame. If you're in the middle, you can tell what frame it is. Don't expect to see that.